Bismillahirrahmanirrahim. Assalamu alaikum. How are you today, students? I hope everything is going well. I would like to bring to your attention to some outstanding discoveries of scientists in plant genetics. Probably you have heard about jumping genes. Do you know why it is called jumping genes and by whom it was discovered? Would you like to know who is the grandfather of genetics? If you haven't heard, let me call their names and their outstanding work and contribution to biology and plant science in my following slides. So let's go look at them. So on the picture, the portrait of Gregor Mendel, the father of genetics. He grew up with a passion in mathematics and science. At his young age, he was fascinated by how things operated and how nature acted, so he worked on his family's farm until he was awarded permission to attend a special high school. After completing his secondary school, Mendel went to the University of Vienna, and he performed a variety of activities while at school to help pay for his tuition and living expenses. So Mendel eventually became a monk in the monastery situated in what is now they call it the Czech Republic. So during his time at the monastery, Mendel had the fascination with how different characteristics of plants were passed on from generation to the next generation. So this process is usually known as heredity. Heredity is the passing down of genetic information from parent to offspring or to the progeny and is the basis for the science of genetics. Mendel's favorite plant to work with was the garden pea. Why? Because for many reasons like easy accessibility, and this is considered as annual crop, and this plant is available also in Oman. So by carefully observing his garden, he identified 7 different characteristics as outlined on the slide, such as seed coat color, seed shape, seed color, pod color, flower position, pod shape, and plant height. On a given pea plant, the pod and seed color were either yellow or green, the flower color was either white or purple, the seed shape was either smooth or ringed. The pod shape was either full or pinched. The plant height was either tall or short. The flowers were found either at the tip of the branches or all along in the branches. So these all called traits. Mendel looked at all of these traits when describing the characteristics of the garden pea and conducted a long experiment that lasted for 7 years. The process of combining hereditary or genetic information from flower parents to form seeds is called fertilization. In fertilization, what happens is pollen, which carries the male reproductive cells, is transferred to the stigma. The female reproductive counterpart in flower and the creation of a seed is initiated. In both plants and humans, the first cell to be formed as a result of fertilization is called a zygote. The first group of multiple cells is called an embryo. In humans, an embryo grows by adding cells, thereby becoming a fetus and then a baby. In the plants, the embryo also accumulates cells, but then becomes a dormant seed, which when germinated, and it starts to grow into seedling. The earliest stage of fertilization in plants can be compared to the same process in humans. So in each generation, the seeds of Mendel's pea plant variations produced plants with identical characteristics to the parents. The plants that produced trait type of seeds were called true breeding, because the traits could be dependent on to maintain the same from generation to generation. The resulting seeds from fertilization will grow into a plant with genetic material from both parents. Flowers are found on the same plant is a true breeding plants, having a male and female reproductive organs on the same flower or on the same separate flowers. On 2 separate flowers, So Mendel's curiosity of the true breeding pea plants led him to hypothesize that What would happen if he took pollen from one true breeding plant and applied it to the stigma of a flower with different traits? So the process Mendel used to combine the hereditary material of one plant with another plant is called cross-fertilization or crossing. When pollen from a true breeding plant was crossed to pollen from a second plant with distinct features or distinct characteristics, Mendel discovered that some crosses always produce in offspring or in a progeny with characteristics that were both similar to and dissimilar to those of either parental plant. So he found that these characteristics consistently appeared in the same proportions or relative numbers. So Mendel wondered what would happen if he combined the many sorts of pea plants. He wanted to see if pea plants emerging from crosses would show new traits, right? So meaning traits not observed in either of the parental plants, because offspring traits were thought to be caused by the mixing of genetic material from the two parents. If the progeny or offspring of this cross-fertilization had traits such as lavender-colored flowers, for example, mixing of white and purple, then Mendel could conclude that the parental traits were passed on equally and blended in the offspring. Instead, what he found, the offspring had flowers that were always either white or purple, but never lavender. As a result, the traits were not mixed or blended. So this observation led Mendel to conclude that the trait of certain form, for example green seed color, was dominant to another form would mask or cover the characteristics of the recessive form. The term dominant and recessive refer to different forms called as alleles. So in Mendel's time, traits were believed to come from unknown hereditary material. So we now know that this material is a gene. In humans, animals, and many plants, there are two alleles for each gene. Through many crosses, Mendel came to the conclusion that the 7 traits could be traced to one gene each, made up of 2 alleles, which are either dominant or recessive. We will talk about this topic in the following my lectures. According to Mendel, if a plant has either one allele or 2 alleles of a dominant form of gene, the plant shows the characteristic of the dominant allele and never of the recessive allele. However, if the plant has 2 alleles of the recessive type, then the plant shows the characteristics of the recessive allele for the given trait because no other form of the gene is present. The state of having both dominant and recessive allele of the same gene is called heterozygous. The state of having 2 of the same type of allele, either dominant or recessive, is called homozygous. By analyzing the characteristics of offspring and diagramming the crosses he made, he was able to determine which 7 of the 14 traits were dominant and which 7 were recessive. Mendel concluded that tall plants, yellow seed color, green pod color, round seed coat, smooth pod, purple flower color, and flowers located all along with the stem are dominant forms of the respective trait. Based on his observations, Mendel could make 2 generalizations about inheritance or the passing down through generation of traits. These generalizations have come to known as Mendel's laws of heredity. The first law, unit factors exist in pairs, always in pairs. The second law, in the pair of unit factors for a single trait in an individual, one unit factor is dominant and the other is recessive. The third law, the paired unit factors segregate or separate independently during gamete formation. So we will, in my following lecture, I will talk about in detail the formation of gamete and how many stages. The first law, traits assort independently during gamete formation, and that all possible combinations of gamete will form with equal frequency. So even though Mendel made many brilliant observations, the meaning of his work would not be known until 60 years later, in 19th century. The three scientists from Europe, named Correns, Tschirschmark, and Rischredish, covered Mendel's published articles on his pea plant experiments. The exact experiments they performed in the same crosses he had performed more than a half a century earlier. Those scientists found that Mendel's results to be reproducible, and they published their own article in scientific journal later. So giving credit to Mendel for his discoveries from decades before, the scientists brought attention to Mendel's work in a new scientific era, in an era that would be accepting for his ideas about heredity. 30 years after the discovery of the Mendel's law, and a century after Mendel carried out his experiment with pea plants, another young plant geneticist from the United States was Barbara McClintock. She was studying chromosomes and microscopic structures made of strands of genes from corn. McClintock was interested in studying the structure and behavior of the chromosomes in the nuclei of corn cells. Using corn as a model plant, McClintock was the first to discover transposons, which are genetic elements that can move about an organism's genome. A complete set of chromosomes contain all of the genes. Transposons are not found in every organism, but they are common in corn. As she is holding in her hand, if you also have seen the Indian corn with a mosaic of colored kernels, which is colorful, then you have seen transposons already. Transposons, or in another slang name is jumping genes, are small genetic elements that may contain an entire gene. Transposons get their name from their ability to undergo transposition or movement to new location in the genome. So we will once get back to this topic containing transposons, what is transposase and what is retrotransposons in detailed explanation in my following lecture. So one thing I want to make sure that don't be a cheater like a transposons during the exams. McClintock discovered the mechanism of mobilizing genes within a genome, but since transposons didn't follow the Mendelian law of inheritance, there were many skeptics about her work, making her finding hard to impress upon the male-dominated scientific community was the discrimination against women that is very characterized in the entire nation throughout most of the 20th century. However, she received the highest award from the Genetic Society of America and served as executive boards of many scientific organizations. The McClintock's work was not taken seriously by the larger scientific community until decades later. Social barriers that kept women from reaching tenured positions at universities fueled the scientific community's rejection of her ideas. It was not until later when male scientists studying bacteria also found evidence of transposable elements. And then McClintock's work on transposons in maize was given the attention that she deserved. Since research on bacteria was deemed so important, in fact, that transposons could be found in this organism was extremely exciting to the general scientific community. McClintock soon received an award and became elected to several high posts and prestigious scientific societies. It was not until 1983, 50 years after her pioneering research on transposable elements, McClintock was given her highest honor. She was chosen as a recipient of the Nobel Prize in Physiology or Medicine in 1983. Mendel and McClintock are alike in that the impact of both scientists on the scientific community were not recognized until long after they did research. Mendel was far ahead of his time, and his observations have been fundamental to our understanding of genetics today. McClintock's pioneering work on maize chromosomes and transposition has been invaluable in the field of cytogenetics. Mendel's observations on the genetics of pea plants explained how alleles of genes can recombine in different combinations upon fertilization. The assortments of the alleles among the resulting gametes or cell sex is random, but the location of the alleles is always at the same position on the chromosome. Using corn chromosomes, McClintock studied the mechanism in which genetic elements move about the genome in a random fashion, leading to new chromosomal composition with genes in completely different locations than those found in the parent or previous generations. So this is all for today, and I really hope that you have picked up some useful terminologies from my lecture today, and this will be very useful also for your future activities in my following course. Thanks for your attention, and I will see you then. Bye bye.